It is October 26th, and today we will be focusing on cell structure and function. And one of the observations I'd like you to make about the board are some parallels in the heading of our notes today and in our student will be able to. So we're trying to upgrade our vocabulary from middle school to high school. So when we talk about structures of the body, I'd like you to start using the word anatomy. And when thinking about functions of the parts of the body, I'd like you to start using the words physiology. So starting to make these connections and upgrading your language. And in reference to our STAR question, some of you might be able to answer this question, and some of you might only at least know what the second part is. So let's just start there, right? How is a cell like a factory? What is a factory? What does a factory do? It makes things, right? So it produces some kind of product. So then what does that obviously tell you about the cell and the purpose of a cell? The cell probably reproduces or produces something or makes something, right? So cells, the primary function of a cell is to produce proteins. Proteins being the product of our factory. Right, so writing your answer to this STAR question as well. This is a major foundational concept that we're going to revisit throughout the year, that cells produce proteins as their main product. So, Alvin, did you get us started off with our quote reader, please? All right, so Alvin's going to pull someone to read our quote of the day, and maybe they can even say a little more about it. Is that what's right here? Yeah, I think she's going to be in here. Hello. All right, if you could read our quote and perhaps say more about it.
Body cells replacing cells as you know, everything you know tells them there's nothing more than I think it's about
All right, great. So she's kind of making this connection that when you get cut, does it just stay as like a gaping wound for the rest of your life?
Yeah.
Just bleed continuously forevermore? No, right? What happens?
It heals.
It heals, right? More cells get built. What is some day-to-day evidence we have that you're continually losing cells off your body? What is something visible you can see? All right, so someone said skin. Please, say more. How do you know that you're losing skin all the time? All right, so you're taking dryness, you're making good connection here, we're getting there. People who have a darker skin tone can see this easier than those with a lighter skin tone. Yeah, so say more. So when you're seeing ashiness, what are you seeing there? What is it? It's not just dry skin, it's actually skin cells that have died and are flaking off, right? And this happens with every human, it's just more visible contrast if your skin's a little darker. Now, what this means is that you're constantly renewing and replacing cells that have been lost. And let's say you don't see someone for a few years, you're seeing a completely new set of skin cells than they had last time. You're seeing a reproduction, you're seeing a memory. Yeah?
You know how like for us, like if like we get cut or something, like we heal and stuff. I heard that like if you have diabetes or something, that like you don't heal as fast.
Right, so there are certain disorders where your cell renewal isn't as strong. Hemophilia, right, being one that like your body doesn't close up, right, so you just continue to bleed. And other disorders might prevent your cells from getting the nourishment they need, and therefore their repair cycle is slower. Right over there. All right, so, and in fact, the average human body is completely a new body every 7 years. Right, by that amount of time, you've got an entirely new set of bones, a new set of muscle tissue, everything has been replaced. Right, now for today's homework, we're going to try something a little new. So in order to combat the low capture rate of homeworks in the past, rather than ask you to write down the questions. I've written the questions for you and put them on this worksheet. Now, what I don't want this to become is a handicap. So if you lose this, where do you think you can find the questions? Right on the school's website, right? So like, you know, if this gets lost, you don't have the excuse of saying, oh, I lost the paper, I couldn't do my homework. You can still find these questions online. This is just to give you a head start. Tonight you're doing the compartments essay side, and then tomorrow night you'll do the second section, which is molecular movement. All right, and at this point, while we're finishing up, please make sure that you open up your table kit and take out all the colored pencils available to you. You need the entire rainbow today. That includes 2 blues, a dark blue and a light blue, and 2 greens, a dark green and a light green. And you'll find those things in there from the previous classes. So pulling out red, orange, yellow, 2 greens, 2 blues, purple, and brown. Are we doing some steel tricks? And again, these things were present in your table kits last period, so they should still be there now. You'll see later on. Alright, so again, removing the whole rainbow, 2 versions of blue, 2 versions of green, violet.
I think it's right.
You'll see it right on.
Yeah, they moved it. I don't have no red,
Alright, you should have 9 colors when you're done. You'll have,
you had
you got
0
brown.
out of 2. Come on, babe. Ew, there's still scavenger thing. Look.
The whole rainbow.
Eddie, I got it.
Alright.
Red, orange, yellow, green, blue, violet, and brown. I think you guys have all. And if you're missing purple, just use the paper. All right, terrific. So we're just about there. We've got 5 minutes left. Let's get out of here.
Does anybody need a green? Alicia, give me a red.
All right, folks.
We needed that.
Yes, that was
Okay, pass it over here. We need that.
Here's our folks. And folks, there are also our pencil sharpeners in your table kit. Right, there's handheld ones. Of course. Follow the words in. Just like you wrote the answers down. All right, folks, let's move on. Pencil sharpeners. I'm sure they're good enough. taking so long? The last classes all did it within a minute. So people return their seats so we can get going, please. If your tips aren't perfectly sharp, it's okay. You're not doing that much coloring. Just underlining some words. All right, moving on. All right, so our first word of the day is plasma cell membrane in red. And as always, we're going to look at the vocab and start to add some deeper meaning to the vocab as we go along. So let's start off with this word plasma, which is going to appear in several words today. So anytime you see this word plasma, we are referring to some kind of fluid-like substance.
What color is
So red, please. So plasma is a fluid-like substance, and we're looking at this part because it's going to appear in several other words today. So let's start off right. Now, cell membrane, that's familiar as well. Where have we heard that before? Remind me from the kingdom definitions we did. What was something folks might remember about the cell membrane in reference to the drawings we did or the conversations we had? Protection, right? It's an outer boundary of the cell. Terrific. And you'll notice that I've done the trick I've used before about separating a prefix and suffix of a word. When something is semi, what are we saying about it? Half of, right? I've got it with parentheses here. So when we say something is semi-permeable, we're saying that it is half permitting or partially permitting. So we're saying that this boundary lets some things in, but not everything. Why do you think that is? What are some things that a cell might want to allow into itself? Thinking about a cell in your body. What is something it would want to accept from the bloodstream of the environment?
Oxygen.
All right, maybe proteins like nutrients or gases like oxygen. Right, so the cell is going to allow certain things to enter the cell. What are some things that could possibly be in your bloodstream that your cell might want to keep out? Tamara, you want to help them out? I think I heard you say a good one. Bacteria, right? Maybe there's something invading your bloodstream and your cells might prevent access. So we kind of think of this membrane, right, as like the security of a building, right? It's only going to allow certain things to enter and exit. All right, and next up, we can talk about our cytoplasm. We're doing that in yellow today. The prefix cyto, which will appear throughout the year in several different vocabulary words. Cyto means cell or cellular or some kind of small space or chamber, right? So cytoplasms and plasm we covered before, right? So we got plasma up here as a fluid, so we're talking about a cell fluid. Now this substance maintains the shape of the cell, right? So it fills the inner volume, it fills the inner space of the cell. And we can kind of think of this as being analogous to a water balloon, right? Your membrane would be the balloon, the rubber of the balloon. The water would be your cytoplasm, the fluid that has the balloon hold its shape to a certain extent. If I were to pierce the balloon and let all the water out, what would happen? It would deflate, right? It would become flat, right? So without the cytoplasm, the cell would not have its three-dimensional shape, right? It might be just more two-dimensional. Let's go back to our idea of a factory. So we're going to build on this concept throughout the class. So if we're saying that our cell is like a factory, what would the membrane be?
In the middle.
Would it be in the middle or?
The fence.
All right, so it would be the fence of the walls, right? So we could put, right, this would be like our walls. Now we also talked about how it lets some things in and some things out. So what would be access points of the walls of our building? Things that something could come.
Bodyguards.
Or you say guards or, doors, windows, whatnot, right?
Security system.
We're awesome, right? And your security system would be against the doors, right? It would allow certain things to come in or out or sound the alarms. Right, so we're kind of building on this concept of the cell as being a factory. So then what would our cytoplasm be? So we've got our walls, and then the cytoplasm would be what? In between the ceiling and the floor, all the space inside, right? So, and we could call this the workspace, Right, so we're thinking of this as being where the place where everything inside the cell is going to happen. Now, if you're a prokaryotic cell, you have a membrane and you have a cytoplasm. So, but if you're a eukaryotic cell, what else do you have? Something that prokaryotes don't. So prokaryotes and eukaryotes have membrane and cytoplasm. But what do we need next? And the person is going to get pulled. Rayana, if you could have that popsicle stick for our next contender on our show. Amina, and she's great. Amina, the next object in our tour of the cell is something that eukaryotic cells have and prokaryotic cells do not. It's a membrane-bound organelle that contains the DNA of the cell.
Nucleus.
Thank you, Anita. Could you kindly echo that, dear?
I didn't hear anything.
Oh, terrific. So you still have an opportunity to redeem yourself. So what would be this object within our eukaryotic cell that contains the DNA, right? So we have this large round object with a smaller one inside. It's just on the test we had on Friday. The center of an atom is also called this, if you remember your atomic model. It is the nucleus, right? So in orange today we are going to do our nucleus. So the nucleus is the control center of our cell.
What color, man?
And it can see orange, please. And so this control center holds all our DNA code.
I don't think I'm gonna
And as we're writing this, we're going to add more to this sentence to make it a little more complete and rounded off. In our start question, what did we say the primary focus of our cell was in reference to production? What is the cell trying to make? What,
produce protein.
proteins, right? So this DNA code is for proteins, right? So the nucleus is going to instruct the rest of the cell how to make these proteins. And again, if you're a prokaryote, you still have DNA, which codes for proteins. It's just that the DNA is loosely bound in the cytoplasm instead of inside a membrane. All right, so let's talk about the organelle that is going to produce these proteins for us. And that would be our ribosomes. So in the darker of your two greens, using your darker green, you're going to underline, shade in, highlight the word ribosomes. And ribosomes synthesize proteins. Some of you might be able to help us and give us access to this word. What is it to synthesize something?
Copy and paste.
Kind of close, you're getting there.
Make it bigger, like exaggerate.
Just the first word, what's the verb?
Oh, the same.
One said you started your sentence off with it.
To make.
To make, right? So when you synthesize something, you are making or building or constructing or putting together something to that effect, where you are taking materials and you're using those materials to make something new, right? So whether you're synthesizing, you are making, building, putting together. And so let's think about this in the lens of our factory model again. So in our factory, what would the nucleus be? If we're saying this is issuing the orders, it's the control center, it has all the designs, the blueprints. What role would this be? What person?
The boss.
That would be the boss, right? You stare at the clock already? We've still got like 45 minutes, girl. We're gonna be here forever. All right, so this would be like the boss, right? And if the nucleus is issuing orders to the ribosomes, what does that make them? Those are our workers. Right, so we're thinking about how the ribosomes, they're the ones following the orders of the nucleus. They're going to be combining all the molecules into making proteins. And we'll look more deeply at that process in chapter 12 later on this year. All right, great. Looks like folks are ready to move on. All right, so in any factory, you've got your workers, and where are they doing their work in front of? Or what are they using to do their jobs? There's machines, right? And then there's that long stretch in front of them, like things are coming to them and going past. Awesome, right? And that, in this situation, is our endoplasmic reticulum. So using our lighter green, and as always, let's make some meaning out of the separate parts of this, right? So we have endo, meaning in or inside. What was plasmic again? Can folks remind me from the previous slide?
Fluid.
Some kind of fluid, right? Very nice. And then reticulum is a little less common. Its root words indicate that it is some kind of hall or passage. Right, so this is a hall or passage, filled with fluid on the inside. And it's the place where the workers are creating their proteins, right? So here in the endoplasmic reticulum, proteins are packaged up, right? You can imagine the workers putting things inside the boxes and sealing the boxes up. Move along. One thing is to stop and wait. It's one whole thing to keep going for like 5 minutes, right? All right, so it also synthesizes lipids. And has anybody seen this word before? And know what's the more common everyday language for it?
Lips.
Or you think of lips, right? Sure. And this is a highly concentrated caloric food source, one that you want to avoid having too much of in your diet.
Sugar.
All right, sugar might be, what sugar is a carbohydrate, so we're getting close. It's fats. Lipids are more high school way of saying fats, and we'll talk more about those later on. You'll also talk about them in health with Ms. Kacharian. All right, great, so we have our endoplasmic reticulum, which packages proteins and synthesizes lipids. And next up, we have our mitochondria. So today we're doing this in purple, so getting your purple pencil or marker, whatever you're using. And mitochondria produces energy for the cell, right? So this is the place where we are going to power the organelles that are going to produce and transport the proteins. And a big part of what they're doing is they're breaking down sugars, the main source of energy for the body. And do we have any folks in the room who watched the first 3 episodes of Star Wars? Like episodes 1, 2, and 3, where like Anakin's a little kid up to a teenager? No Star Wars fans in the room? All right.
Don't lie.
So since there's a new film coming out for that, you might want to visit those films, to equate yourself with the characters. So in that they talk about these fictitious organisms known as metachlorians, tiny little microorganisms that give Jedi the power of the Force. Very similar to how mitochondria produce power for the cell in our bodies. Now there's some special things about mitochondria that we want to highlight today, which we'll explore more deeply later on in chapter 10. But for now, we're just going to make some quick notes about it. For one thing, mitochondria have their own DNA. So this is something unique to them as an organelle in an animal cell. Honors folks, a couple weeks ago I had you guys look up a word that talked about how prokaryotes began to live inside some eukaryotes, and they exchanged energy production for shelter. Do any of you honors folks remember what that word was? When one organism was moving inside of another, and then they worked together to survive better? We even saw that prefix inside today, perhaps on this very slide. Endosymbiosis. Right, so the evolutionary theory is that mitochondria are once their own individual organism, a small prokaryote. They took up residence inside a eukaryotic cell and exchanged shelter for the production of energy. And this is why we believe that they have their own DNA, right? It's a vestigial structure from their ancestry. And another interesting thing is the mitochondria you get are from your mother, right? So sperm do not have mitochondria, but egg cells do. So all the mitochondria you have were donated by your mom. All right, so let's move on. All right, now, after a long day of producing products at our assembly line, what do we expect the workspace to be like? Just like this building at the end of the school day.
Empty.
Well, the students all leave, and what's evidence that they've been here?
Mess.
There's a mess, right? So messes get made in our cells as well. Right, so in our darker blue, we're going to talk about peroxisomes. So taking your darker blue and shading in or highlighting this word, underlining, or putting a dot next to it. And is this word familiar to folks? All right, so Diana's thinking of peroxide, right? A common household disinfectant. Right, so very similar here, right? We're talking about a chemical that's going to cleanse the cell of particular toxins. Right, so the peroxisome's job is to help maintain the homeostasis, the hygiene of the cell. And once our cell has done some of its tidying, done some of its cleaning, it has a new problem now, right? It has a lot of structures that may or may not need to be taken away completely. And that's going to take us to our lysosomes, which we'll do in lighter blue. Let's go ahead and take this prefix off and discuss its meaning. Whenever you see the prefix lyse, not L-I-C-E, like the arthropod that might get into your hair. This is lyse as an L-Y-S, and this means to cut, or to sever, or to splice, right? We're talking about some kind of opening is being created or something is being separated here.
What color is that?
Our lighter blue, please. And just like our peroxisomes, our lysosomes contain chemicals but instead of, for the purposes of hygiene and cleansing, these are more about taking away things that are broken and unusable. So perhaps you have a ribosome that is damaged and no longer producing proteins, and you need to get rid of that ribosome. The job of the lysosome would be to release chemical enzymes to break down and destroy that. Let's continue with our factory scenario here, right? So we've got the role of one person who's supposed to tidy and clean this space up. So our peroxisome would be like what? Yeah, it would be like our custodian or like our janitor, right? Right, so, and what about so our custodian's gonna sweep everything up, kind of tidy the space. What is our lysosome gonna do then? So we're saying it's taking away all the bad stuff, all the broken stuff that we don't want in the cell anymore. Get closer. Who removes our waste? At the local level, but we're saying like takes it away completely. Yeah, right? Right, so you kind of think of this in a loose sense, right? And we're talking abstractly here, of course, not strictly. But this is kind of be like your garbage man, right? It's going to take away all the broken things that are no longer needed or necessary for the cell. It's going to dissolve them or remove them. All right, terrific. Let's move on to our next slide. All right, Anne. Next up in our brown, using our brown colored pencil, we have the Golgi apparatus. Yes, Amina? Yes, please. Yeah, go for it. Right, so taking your brown, we're talking about the Golgi apparatus. Now, the Golgi apparatus, its job is to transport proteins around the cell intracellularly or external to the cell extracellularly. And of the words we've talked about so far, which of these things you might imagine this one is attached to? So which other thing has to do with like the movement of proteins, moving them in and around the cell? The place where they were packaged before? Excellent, right? So we can kind of think of the Golgi apparatus and Endoplasmic Reticulum as having an association. And you'll see when we get to our drawing that they look similar as well. Now, at the end of our workday, are we going to have used up every material we have to start with? No, right? So it's like if you bake something, you use the whole bag of flour and the whole bag of sugar every time? No, so what do we need to do with those materials?
Save them.
Save them and store them for later, right? And that is the purpose of a vacuole. So many cells, eukaryotic cells, have a vacuole. That's just in our normal pen, pencil, whatever you got today. And the purpose of the vacuole
Excuse me.
is to store reserved water, to store reserved dissolved salts, minerals, nutrients, whatever the cell might need for later. And the vacuole is a really good way to observe the difference between two major cell types we'll be studying this year. What kind of cell, what kind of kingdom's cell do you think would have really large vacuoles? What kind of organism would need to store a lot of water? Plants, right? And why is that? Why would a plant be more concerned with storing water in its body than an animal would? What are something animals can do that plants cannot?
Photosynthesis.
So plants are doing photosynthesis. They do use water as a fuel for photosynthesis.
They can't like go out and look for it.
Right, so animals can move to the place where there is water, but plants have to make do with what's available to them at the time. So they might store it for later on in their vacuole. All right, terrific. And let's go ahead and finish off our factory analogy. I've specifically chosen brown for Golgi apparatus to maybe give you some real-world meaning here. Who do we think of when we think of transport? They're dressed in brown and driving a big brown vehicle. UPS, right? So you kind of think of these, right, as our shipping, as our UPS, right? Our Golgi apparatus is going to package up and transport the products of the cell. And then, of course, again, thinking about our factory, what's a place that we could potentially store things we're using later on, materials for later?
Storage room.
In a storage room or something, right? Or in a basement or whatnot, right? So this is just your storage area. And we can put, shed or basement, whatever you like. All right, what we're going to be doing next is we're going to be drawing a diagram that matches our notes, right? So we'll be color coordinating our membrane to the membrane color and our mitochondria color. But before that, we're going to have a small video presentation. And some of you may have seen before, or maybe some other weird stuff is happening from before. Yes. Yeah, I'll send you in just a sec. All right, so, and please don't put all the blinds down. It's only a 3-minute video. I think it's a little much. Extra. All right, so let's go ahead and watch this short presentation, then we'll do our drawing.